warmly welcome all to another interview of a series of interviews with eminent scientists conducted by the Department of Chemistry, Faculty of Science, University of Colombo. I'm Sandhini Gunaratne.
And I'm Donisha Pereira. We are final year undergraduates of the Faculty of Science, University of Colombo. Today, we will be interviewing Dr. Binodh DeSilva, who is the Vice President, Lead, Discovery and Optimization of Small Molecule Drug Discovery at Bristol Squibb, New Jersey, USA. She received her bachelor's degree from the University of Colombo, Sri Lanka in 1987 and a PhD in Bioanalytical Chemistry in 1994 from the University of Kansas. She was a postdoctoral research associate in the Pharmaceutical Chemistry Department at the University of Kansas from 1994 to 1995. Dr. Binodh DeSilva is a past president of the American Association of Pharmaceutical Scientists. Also, she is a member of the American Chemical Society, Association for Women in Science, and the Healthcare Business Women's Association, New York, New Jersey Metro Chapter.
Good morning, Dr. Binodh DeSilva. Thank you for giving us this opportunity and time from your busy schedule to take part in this interview. As an introduction to our discussion, Dr. Binodh, we would like to know a little bit about your family background, early education, and also the inspiration for entering into the field of science.
Yeah, so thank you very much. I mean, this is a great honor to be with all of you, especially as an alumni of Colombo. And this is, I mean, I've seen some of the videos before. This is a great thing you guys are doing for the students and anybody who wants to see these things. So thank you very much. It's a great honor. So I'm born to two parents who are science teachers. My mother was a chemistry teacher and my father was a physics teacher. And then my mother continued to teach chemistry, for a long time until she retired. And then my father, of course, was a physics teacher who, became a principal after that. So we kind of knew what it was to be in the science. And, at home, it was a school, right? A principal, a teacher, and students. But having said that, I have a sister and a brother younger to me. Even though they were both science teachers, they never forced us to go into science. So, I'm the only one in science. My sister is a lawyer and my brother is a marketing, management, marketing, sales graduate. So as you can see, they were never forced us to be in science. And then most of the inspiration and everything came from school, right? So I was at Visakha and the teachers were very good about, guiding you to doing different things. And also then you get into the sciences and then you enjoy what I really enjoyed was really understanding a lot of the chemistry behind things. And I really loved botany and zoology. So really getting into the scientific aspects of that's kind of what part of my inspiration was. So, yeah.
Great.
Madam, you have obtained your bachelor's degree from the Faculty of Science, University of Colombo. Can you tell us more about your undergraduate life and how it helped to shape your career and life?
Yeah, so Colombo was great in that there were a lot of great faculty members and everybody, right, that would teach us. But I think one of the things that I still remember was a professor, specifically Madame Pereira, who was I think she was either head of the science faculty or dean of, I think it was the dean of the science faculty. I think she's probably the most inspiring person. Oh, I shouldn't even say that. She's the one who got me into chemistry. Let's put it that way. So when we were, I think it is the same way. You do 2 years of 3 subjects, and then you then get into special degrees, and then you go into, right? So I think it's the same way now. So when I got into the special degrees, you know how they put these things on the bulletin board, you know, who got selected to the different special degrees and such like that. So I got all three. I could do all three, botany, zoology, and chemistry. And then when I looked at the chemistry list, I was the only woman. The rest of all male colleagues. And so I'm like, oh, I'm not going to deal with these guys. I'm just going to go do botany. So I was going to go, at that time, the prominent professor at that time for us was Dr. Kotagama, I mean, who used to be at Singaraja doing all kinds of very cool research. Right? So I was all ready to go into research with him. And then I got a call. I mean, I was summoned to the dean's office, and Madam Pereira sat me down and gave me the Rights Act and said, oh, well, you know, there's nothing more central than chemistry. And what's your reason for going into botany? So at that time, you know, you guys are final year students, so you know, we kind of, you know, don't really know, you don't have the wisdom at, you know, 50 plus years old, right? I just blurted out, I'm not going to deal with those guys. I just, you know, I'm the only woman there. She just let it on. And then that's history. So I was in chemistry then. She said, no way, you're not just going to quit it because you have to deal with these boys. I'm like, okay. So I think that's kind of how I mean, that was probably, I would say, the turning point for me to get into chemistry. And then my professor in Kansas was who kind of guided me. Yeah.
You have studied bioanalytical and pharmaceutical chemistry for your PhD and postdoctoral work. Why did you select these fields to specialize in?
Yeah, so that, just like I said, my PhD advisor is probably the one who directed us into Bioanalytica. So my undergraduate research thesis was with Dr. H.D. Gunawardena in Analytica. So that was what I did for my undergraduate research. And then, so being in chemistry and in analytical chemistry, it was really important to see how these things matter in day-to-day life even. I'm not sure what you guys call it, the industry internship or something that we get to do. I was at the government analyst department too. So then you get to see how important analytical chemistry is, right? So you really get to figure out what's going on. So I kind of like that area. Then when I came to Kansas, my professor, Dr. George Wilson, he's an electrochemist, and I knew I wasn't going to do electrochemistry by any means. But looking back, I did not realize it at that time, but, looking back, almost 20 years ago, he was such a visionary. He told us, all of us who were joining him told us that there's nothing in the future that's going to be pure chemistry. So just go to anywhere you want and just learn some life science and combine that with chemistry. Because he said, do whatever you want to do. So that's what led me to go to Bioanalytical, right? So I had a bio background, but then we were told to go take classes in molecular biology, immunology, biochemistry. Everything, he told us just go take these courses, and then pharmaceutical chemistry. Pharmaceutical chemistry is a different department than chemistry in Kansas. So I did courses in pharmaceutical chemistry, learned the chromatography and all that there, and then immunology. So I liked immunology. So then I combined the immunology with my analytical chemistry that's how I did the bioanalytical. So bioanalytical chemistry was mainly to really understand how do you measure things, how do you quantify drugs and use in the biological matrices. And that helped me with my career moving on in the pharmaceutical industry as well as in what I do now. I don't do too much lab work now, but that's what I leave. So it was, that was really, really key to understand what he saw was the future to direct us, right? That's just so visionary in some ways. And you don't realize it until you get to a certain stage in life. So listen to Dr. Abhay Singh and them when they tell you something, when they see something that we don't see for the future.
Right.
Yeah.
Madam, as I was going through your research, I found that you have published several papers on bioanalytical method validation of ligand binding assays to support pharmacokinetic assessments of macromolecules. It has helped the FDA to incorporate ligand binding assay criteria into their bioanalytical guidance. Why have you focused more on the criteria of ligand binding assays of possible drug molecules?
Yeah, so one of the things is, so my PhD background, everything was in macromolecules, right? In proteins, in antibodies, understanding antibodies and all that. So then my postdoc was understanding mass transport to solid tumors, but the mass transport was again an antibody. How do you tag an antibody to deliver it to a solid tumor? So, and when I was working, after the university education in the PhD, I went into the industry and I was working on proteins as therapeutics, as drugs. So unlike the small molecules, right? So there's chromatography, mass spectrometry to quantify. There's really no way to quantify proteins and macromolecules. So what we had to do was really use ligand binding assays. So, and protein, this is in the 95, 96, 2000 era, right? Proteins were just coming to be drugs. And you didn't have a way to quantify them besides ligand binding assays. And the FDA, who really governs and regulates the approval of drugs to be marketed, didn't have a way to have guidance for ligand binding assays. So we as a community got together to understand what was needed. So the technique was different, right? And there was no chromatography, there was no separation in ligand binding assays. So there was a huge need. So it was the industry collaborated well. I mean, I wasn't just the only one who was participating in these things. The whole industry got together to understand what was needed to guide these regulations. And you have to have a method validated to determine pharmacokinetics. Okay, so because of that, you really need criteria. So for a method to be said, okay, it is accurate, it is precise, it is specific, you need guidance, you need criteria. You can't just say, oh, by the way, this is what it is. So you really need, for examples, we say for a method to be accurate, it has to have 20% precision, 20% accuracy, against the standard curve and such like that. So that's where the criteria becomes important. And when the industry got together and really, because the industry does the research, right? So then we tell the FDA, this is what we do. And then they can partner with us to say, yes, these are good criteria. Let's see how that can become into guidance. So this is a lot of time. I mean, at least we started in 2003 and the guidance actually came out very recently. So it took us a good 10, 15 years to get the guidance.
Can you elaborate more on how bioanalytical method validation of ligand binding assays can support pharmacokinetic assessment of macromolecules?
Yeah, so you guys know pharmacokinetics. It's really what the body does to the drugs, right? So the ADME properties, the adsorption, distribution, metabolism, excretion. So the ADME properties of a drug is so important, right? That's what defines half-life. So like if you take a Panadol, right, the half-life is about 4 hours. And that's why you have to take it every 4 hours to have a steady state concentration in the blood. So pharmacokinetics is the measurement of the ADME properties. So when you have to have good analytical methods to determine those ADME properties, right? So the concentration measurement of when you take a pill, what it does to the body and what the time points and the timeline of the concentration is so important to determine the pharmacokinetics, right? So for a protein molecule, just like a Panadol or something, that you can use mass spectrometry to measure the concentration. So the proteins really need these ligand binding assays, because you can't, I mean, now mass spectrometry has increased and improved, the technique has improved to measure proteins as well. But that's where the ligand binding assays came into play, to quantitate the drug concentrations in serum, in blood, in urine, to determine the pharmacokinetic properties. So that's how all that things then gets, you know, linked in with how you do. So then once we have the PK curve, then, you know, we determine the half-life, the absorption properties, and such like that. So in a similar way, you can also use ligand binding assays to measure proteins and macromolecules in tissues and other biofluids. Does that help? Does that,
Yes.
you know,
Yes.
help? And stop me anytime, okay? So.
Madam, I found that you have published more than 50 papers. This research was done in different areas under immunochemistry, immunology, oncology, and pharmacology. What drives you into these different topics?
So, I mean, that was totally based on the research at that time. So I had different industry experiences, so each company will have their specialty in the disease areas that they are working on, right? So immunology, you know, oncology and such like that. So that's what drove to be in those different areas. But if you really, you know, in answer to the previous question that you had, pharmacokinetics, any drug that we develop need to have the pharmacokinetics. So based on where we were using those methods is where the research was. So that's where we are now. I mean, most of the immunology drugs, most of the oncology drugs are protein molecules. So that's kind of where my research was, based on which therapeutic area we were supporting at that time.
Is there any interesting research area you're working on these days that you'd like to share with us?
Yeah, I mean, there's a lot of things that we are doing right now. So I know I can't go into the details because of, you know, this is being done, right? So I'm right now, where I am today is in the very early research. We don't even have a drug, right? We have a target. We think we like to work on this disease, and this is the target that we want to develop the drug against. So that's the area that I'm in right now. And we are working on cardiovascular, immunology, oncology, fibrosis, and immuno-oncology. So those are the fields that currently BMS is focusing on. So yeah, I mean, we are working on these things. So I can't go into the target details, but it's very interesting because when I started my career, I was like in the middle of the drug discovery process. And then I went to manufacturing, and then I came back all the way to discovery. So now I have covered the entire drug development process. It takes about 15 years to develop a drug. So I'm at the very front end now. So, you know, I've seen about 3-4 drugs that I worked on develop, but, you know, most of them fail. So yeah, I mean, I can't go into too much detail, but that's where we are in the fields of operation for us right now. Those are the areas.
You have worked in several pharmaceutical companies. What are the experiences that you have gained from joining these companies?
It's actually a very, very, very good question, and a very interesting question. You know, we should have a conversation when I come to Sri Lanka one time, maybe, because it's really loaded, right? So I mean, I'll give you the 3 examples that I think helped me. So I work for Procter & Gamble Pharmaceuticals, one of the largest consumer product companies in the world, right? And I was in the pharmaceutical section. Now that was really interesting because P&G, with this massive giant consumer product company, this pharmaceutical division was a smaller group. So it was like being in a small company that was in a large company. So there were good there was good financials there, because finance is very important for developing drugs, right? It takes about, I think right now it's about $1.2 billion to develop a drug. And so that was really good, having that financial, and Procter & Gamble was one of the most innovative companies. And so that innovative analytical technology, the instruments were accessible to all of us. So that was very cutting edge kind of, but at the same time, the culture was very conservative. It's a 100-plus-year-old company, very good culture, very process driven and such like that. Then I went to California and to Amgen. Absolutely phenomenal biotech company, very entrepreneurial. Decision making was very fast, and the scientists were really driving the science and the innovation to get to these protein drugs. Right? That was when the protein drugs were really coming into the market. So that I got that really nice, a wide array of experiences, both scientific and managerial experiences there. And then coming to BMS is probably, I would say, the middle, because P&G was a large company, Amgen was not that large, but BMS is probably about, I think we have about 25,000 people or something like that. So it's probably mid-sized pharma. And BMS culturally is very relationship oriented. So it's really nice to see that collaboration, the cooperation between the scientists. It's really fascinating how that gets on. But all three were extremely innovative companies, but in different ways. So it's that culture and the flavor of each company. Each company has its own culture. I think that's the most fascinating experience that you gain, trying to navigate how can you work in these environments. So, yeah.
Madam, how was the transition to industry after working in academia? What are the difficulties you had to overcome with this transition?
Yeah, so I would say I didn't really work in academia in the sense like as a professor or anything like that, right? So I was in graduate school and then in postdoc. So that's my academic experience. So I didn't, we had to write a proposal to defend, but I didn't have to go get money for a grant and such. So I don't know that part of that academic process. But I would say one of the biggest learnings or the transitions was time management. So, how many times do you tell your professor, oh, yeah, I'll get you that paper. I'll get you that paper. Months pass, right? You don't think about it. But in industry, time is money. So you really have to make sure that you do things on time, and time management skills are so important. And the second thing is the regulation. I went into the regulated side of the industry when I got out of school. And that was a big change because everything needed to be documented. I'll tell you a funny story. When I came to my first job at P&G, I was in the lab. I was, this hot shot PhD, right? I know everything. Going into the industry, and I was weighing my powder at the analytical balance, and I didn't have my notebook near me on nothing. And so I was weighing my powder out, and I weighed my powder, and there was a napkin, like a serviette. I wrote the weight on it, right? So because I can't go write it back. Oh my goodness, there was a 30-plus-year veteran who was in the lab with me. She said, "What are you doing?" I'm like, "Oh, I'm weighing my powder and putting it on." No, this is raw data. You wrote something on the napkin. You need to go paste it in your notebook. You can't just transcribe it. That was my first exposure to regulated world. How many times, in undergraduate, graduate school, that we just write something and go back and write in our notebook, right? So I would say that probably those two are my main, there are a lot of other things, but the main thing about understanding the regulations, because think about it, we take drugs, all of us take, all of us do take drugs, right? These have to be safe and efficacious. So these are highly, highly regulated. for that to happen, we in the lab need to follow procedures properly, even to the extent of writing a little weight down on the notebook, right? Really, and then time. Time is money for industry. It is for the academia too. I mean, you have to get your grants in, the professors need to, but it's a little bit more relaxed than in the industry. So those two probably are my biggest transitions, I would say.
Great.
So.
You have received many awards and accolades, including HBA Luminary in 2018, CPSA Distinguished Scientist in 2017, and Wisdom Share International Mentor of the Year in 2015. Can you tell us more about these awards and how they are helping you to shape your professional life?
Well, you know, you don't really do things to get awards, right? So I mean, I just do what I have to do to help people and do what I do. So I think most of this was really some of the awards were for the scientific innovations, like, you know, getting the papers for validation. I mean, that was a huge unmet need for us. We didn't have guidance for bioanalytical methods for macromolecules. So, you know, working with industry consortia, working with other colleagues in the industry, really bringing something that was not there in the industry to light, you know, that was part of some of the awards that I got. But the Luminary and the Mentor Awards are truly for what I do with the students, what I do with junior people in the organization, Women in Science. Because, you know, if you really think about it, where I am today is because of a lot of people. Now, I didn't come here just out of the blue, right? My parents, what they gave me as values in life. And I was, you know, I was a Girl Guide, I was a President's Guide, and all those experiences, you know, the school. So, and my professors, undergraduate, Dr. Binodh and Professor Tilakaratna, to this day, I don't know if you guys know Professor Tilakaratna. He's here in the US, and we still, I mean, he has what the University of Colombo Alumni Association here in the US. And Sir is, you know, like, I'll talk to him tomorrow, right? So he's guiding us even today. My professors in, so someone gave me a hand. So, you know, part of it is, you know, giving back. So, I mean, I've been doing a lot of work with students. And so that's kind of, you know, what has happened. And I do get something back too. I really get to talk to the students. I understand their needs. I understand what the new research that they do. And all that. It's a both way. It's a two-way street. So, you know, I don't know. You know, I probably get more from what I do than they get from me. So.
Dr. Binodh, you are an active member of many societies and associations, and you are a past president of American Association of Pharmaceutical Scientists. Can you tell us more about this membership and what you have gained by joining them?
Yeah. So, you know, once again, like I said before, it's all about giving back. It's all about society. It's all about community, right? So when I started in graduate school, one of my professors called me and said, you know, can you review these abstracts? It was for a conference. So I started out at AAPS, American Association for Pharmaceutical Scientists, as an abstract screener. So, you know, you know how you go to conferences, you submit an abstract and you had to select the abstracts to be presented at the conference. So I started screening abstracts. So one of probably the lowest level of activity that you can do in an organization. And then I became the abstract chair, and they have communities and they have scientific groups, focus groups. So I kind of joined these groups for not just for me to serve, but also for me to learn, right? So in our research, I mean, all of you know, it's good for us to be participating in scientific conferences and such like that. So with AAPS, I found a home. ACS, I have not been an active member of ACS, but I'm also part of analytical chemistry group in ACS. But AAPS provided me a home to also learn other technologies, other scientific areas, but also to give back to them. So that's how I grew up in AAPS. And there, it's just learning to network, learning other people's research, getting to know students and faculty who do cutting edge innovative sciences. And then it also helps us recruit, right? So for our companies, we get to see the students. So if you go to the conference and read the abstracts and then you follow the student, you can see where they're progressing and if you want to hire them. So that helps us too. And so all that comes into the picture. And then, I'm one of the things I think I have to say, to brag a little is really the whole idea of being elected to the presidency of AAPS. I mean, who am I? I came from this tiny country, Sri Lanka, right, to this country, and AAPS is a national organization. And it's an election process. So someone nominated me to run for president, and the membership really supported me and elected me. So I think I'm really, really proud of that. And then at the same time, everything else. I mean, I cannot forget all the training that I got from Sri Lanka. Right? I mean, when I was a Girl Guide, I was like in the shanties of Dehiwala in Belawata. I mean, all that, I had that service mentality, really supporting the people and all that. And, my parents were early members of Sarvodaya. And, I've gone to the camps, I've gone to the villages with them. So all that kind of brings everything, everything that we do kind of culminates into what we do. So I think I'm really proud of being part of these associations, to learn the science, to learn the innovations, but at the same time to also network and become a part of a community.
Great.
Madam, you have done your PhD and postdoctoral work at the University of Kansas. What were the struggles and difficulties that you had to overcome as an international student? What would you like to say to Sri Lankan students who are hoping to do their higher studies in USA?
Yeah, that's actually, I saw that question and I was like, wow, that's also a loaded question. But I am a first generation immigrant. Okay, so I didn't have anybody when I came here. And so once again, I always have to go back to my beginnings. That's where it all begins, right? My mother was a very open-minded woman of her time. She both my sister and I, she always told us, get a good education, and they provided us a lot of opportunity to do different things. So that helped me. And also, one of the biggest challenges that we had, we didn't, nowadays my husband's nieces and nephews are here, and you guys have the internet, you have FaceTime, you have Zoom. We didn't have any of these things. So to get a call, it was like $3 a minute or something. So it was like you could only call home, just, a little bit of 2-3 minutes. But what you really have to, my advice to everybody is to have an open mind. It is not easy. Right? The first couple of years, first 2 years, you're really getting used to the country. You're getting used to the culture. We whatever said and done, we are a very close group in Sri Lanka, right? We know a lot of people, but our families are really important to us. So to get out of that family environment, this was the first time I kind of lived alone, right? Had to cook, had to do all that. So that's a huge challenge. And you have to be able to do everything and also study, right? So, I mean, it's just one of those things. But I think, one of my biggest advantages is coming to Kansas, although people didn't know where Kansas was at that time when I came. one of the biggest things was Kansas was a smaller university, but also it was in the what we call the Midwest in the US. It was not in the two coasts. That environment was also very nurturing. The faculty, my professor had been to Sri Lanka, India, Bangladesh, so they knew what they were, right? So they was very supportive. Their families were very supportive of us, really nurtured us, gave us that comfort to be there. And then the other thing is, whatever, I mean, you guys do a lot, very well right now, but you have to really pay attention to the language, right? You have to be open because they have an accent. You really have to hone in on those things and be open to that and learn that. Be prepared to live on your own. Be independent before you come, at least for a little bit, so be independent. I think most of you are a lot more independent than we are at this time. The generation gap is really done well. And then you have to be able to manage your money, right? You may get a small stipend to pay rent and buy groceries and everything else. So those are some of the struggles. And then if you come to a place where there is winter, right now, it's winter for us, right? You really have to learn how to manage, getting used to the culture. One of the things that I would really like to tell some of our Sri Lankan students is, this is not typical to Sri Lankan, Indian, Southeast Asian students, is embrace the culture. It doesn't matter where you go, the UK, the United States, Australia, wherever. Embrace that culture. The more that you can adjust and adapt to that culture, the easier will be your time. Don't try to fight it because you're now in another country, right? So if you don't embrace their ways, I'm not saying to embrace the bad things. I'm saying to embrace the good things. Keeping your values, because your parents instilled values in you. So keeping those values, but embracing the culture so that you can really adapt and be flexible to that culture. So I think that's probably my biggest message. And, there are so many resources here. Take advantage of that. The research is just really good. I mean, when you're in school, or you come to do a postdoc or whatever, embrace the resources and the intellect and the innovation capabilities we have here to further yourself and your education. I think that's very, very critical. Hopefully that serves a lot. That was a lot. Sorry.
No, it was so informative, madam. Nowadays, more females are moving into the research field. So as a woman in science, what are the difficulties you found when working in this field, and how did you overcome them and balance your personal and professional life?
Yeah, so I mean, once again, maybe I'm a little different, but because maybe I had a strong mother, and I had a lot of very, very strong role models, women role models. Madam Pereira, and then I had a lot of good, Girl Guides and at Visakha. So I think that maybe that was one way for me to kind of do whatever I want to do. So I was never afraid to do anything new. So that was, I think, one of the things that sometimes we think, oh, and it never, I don't go about doing something thinking that, oh, I'm a woman, I can't do it. You see what, so it's also all mental, right? I mean, it's all in your mind, your mindset of doing something. But there are challenges. There are really, really big challenges because just the way we are brought up, the way we are nurtured, we don't take failure well, right? As women, we think, okay, it's our fault. What's the first thing we say? Oh, I'm so sorry. It's just, you say that, but you've done nothing wrong, right? So I mean, it's just that the nurturing, the way we've been brought up. Having said that, even if you do that, even if you think that, okay, I made a mistake, accept that, right? And say, I made a mistake, here's the next steps. I think that's something that we need to be confident about. And there's a lack of confidence sometimes we place when we are in a situation to say, okay, so now we are in this situation. I know only about half the stuff but embrace that, acknowledge that, and move forward. So I think that's another thing. And so the other piece, which is a little personal, that each woman, each female needs to figure that out. We really, when do you have a family? That's part of life. Don't ever forget that. That's part of life. And it is an important piece of our growth, right? But that's a very individual thing. So what I've seen sometimes happen, and then it's no fault of anybody. No, I'm not judging anybody, but sometimes we as women, we can go a long length, farther out into doing research. But once we start a family, that becomes our main goal. There is a way to be a part of our goal, right? And also be professional. And for that, we have to have an open mind, right? So I can't talk about myself and my personal life. My younger daughter, she calls me absentee mother because she grew up with a lot of nannies and daycare. And because I travel a lot, this year it's been great. I didn't have to travel anywhere, but I travel a lot. So she has always been with nannies or babysitters. I have to be open to that, right? Because she has to do this temporary thing, because if I'm successful, she'll also see a role model, number one, but at the same time, I'm doing all this to give her comfort, right? So, so that's the kind of thing that we really need to think. I really acknowledge and appreciate that it's very personal. It's very personal. That I am comfortable with that, right? But not many people maybe. But there is a way to do both, okay? And then the other piece that is very, very important for women, especially women, we are not perfect. Nobody is perfect, right? I don't care who you tell. All of us have our good things, our strengths, and our opportunities. So because we try to be perfectionists, and then we get frustrated, we get stressed out, right? So, those are some of the things that probably we need to kind of avoid. But once again, it's doable. You need a whole village, right? I'm not saying, when my children were born, my mother was here and, I got all the help in the world. And you have to be open to asking for help. And sometimes we say, oh, I can't do it all by myself. No, you can't. And if you can't wash dishes, tell your husband, okay, you have to wash dishes. You have to be, because it's a partnership. It's, really, and I see that happening a lot more, that, even in the Southeast Asian countries, I've seen the roles changing, and, it's becoming a lot more aware, and, there's a lot more partnership in family life. And so it's coming in the right direction.
Great.
Madam, I read that you are a dedicated mentor and is passionate about supporting junior scientists. Many young people like to work and experiment in science and make research their career. Do you have any advice or message you would like to convey to them?
Yeah, so like I said earlier too, that someone gave me a hand, someone helped me. So that's kind of my goal, to help other people get to where they can be, because everybody has a potential to do that, right? So I do mentor a lot of students, a lot of junior scientists. I think it's better in Sri Lanka than here even. There are a lot of girls who don't go into science, to STEM education. science, technology, engineering, and math. That's a big initiative here. So as early as we possibly can to get people to get into the sciences. I mean, it doesn't have to be chemistry or biology or anything like that, right? Computer science, if you're an engineer, do whatever you want to do. So I think we need to give them opportunities. So I don't know about even you all, right? I mean, if you go back to your high school, to go back to the school that you came from, even if you go back there and tell them it's fun to do science, it's fun, right? Then you get more and more junior people interested in research. I think that's what we can do, and that's what I like to do. I mean, I really like to encourage people to go into research, it's not for everybody, right? I'm not saying research is for everybody, but at least get some people interested. So, that's what I like to do. That's what I do with this. I don't know what message they'll take, because sometimes when we go to between grade, here, at least in the United States, between grade 6 and grade 9, what some schools call middle school, that's the time we need to get kids interested in science, because that's the time they find up maybe not going into science, right? So getting into research. So if you can do small experiments with them, showcase like the science fairs and such like that, that's the kind of thing I want people to get interested in. And you all can do that very well too. Go back to school, your high school, talk to people. I mean, I hope that happens in Sri Lanka now. I don't know that as much. Maybe it does.
Yes, madam, because we have some societies in campus, so we go and do some seminars in some rural areas. So I think it helped them. Yeah, because me and Donisha were part of the Leo Club of the University of Colombo. So both of us worked. Yeah.
Oh, that's nice. That's really nice. You need to. Yeah.
Even the schools are interested about that, madam. They are calling us to do some kind of sessions with students to get them aware of these things.
Oh, that's beautiful. Yeah, that's really good.
The next question, Dr. Binodh, where do you see yourself in 5 years? And what changes do you expect in your field of work in that period?
So I hope I'll be retired in 5 years. I really do. So in the pharmaceutical sector, I would say right now the cutting edge is cell and gene therapy. So I think that's going to be so prominent going forward. The next few years we will see, so right now CAR-T, cell therapy is approved, right? A couple of drugs are approved. Gene therapy is next. I mean, that is the way so now I'm forgetting, human genome was understood, maybe 10, 15 years ago now, but we are still not there, what can be done with gene therapy. So I think those two are, I mean, clearly, that those two will probably be the most prominent. I mean, look at now, right? The technology platforms, even for the COVID-19 vaccine, right? The mRNA platform, which we think mRNAs are really unstable and all that. That platform has brought to life what can be done. So I think that platform is going to take off for other things as well, not just for vaccines, but for other treatments. So as the technology improves, and then from the pharmaceutical side, the gene therapy, cell therapy, and then some of these technological advances will really revolutionize the pharmaceutical industry. On the other side is the data and the AI, artificial intelligence, the data science, right? So we generate so much data, right? And then we really need to capitalize on that to understand the trends, right? So to match patient data to the outcomes, what we call outcomes research, right? So, you have your patient data and you have an outcome, so even with COVID-19, let's look at this way, right? So recently, I think I read it yesterday in the news that, some patients who had underlying conditions had most people didn't have any effects, right? It was fine, it was like the flu shot. But some people reacted because they had an underlying medical condition. So in that case, you can see, okay, these patients got the vaccine, they had these underlying medical conditions, what was the outcome? So those data science with the patient outcome, that's going to also revolutionize the way we treat patients, right? So hopefully you and I, in another 5, 6, 10 years, we can probably get, I'm sure all of you guys have these digital wristwatches, the iPhone, everything, right? We'll get our data, right? Our medical data in that. And then we'll be able to look at it and say, okay, I'm seeing this. What is that outcome? You should be able to go to the doctor and ask, okay, here's what I'm seeing and here's what the data tells me. So I'm hoping that, if I have a disease, I say I have diabetes or something, and I have my diabetic results, and then I'll probably get some cohort between my age group. What is it that they are doing that I can compare and contrast, and what are the treatments options? So I think those are things that I see that's coming, and it's totally going to revolutionize the way we treat patients. And the pharmaceutical industry. I mean, that's the industry that I know of, right? I don't know anything else, but yeah. So that's really what I see. But hopefully, I'll be retired watching it from the sidelines.
As a wrap up for this discussion, I would like to know about your hobbies, more about who you are outside of your research and career.
Okay, so my biggest challenge, doesn't matter what it is, give me any research project, but being a mother. I have 2 girls. One is right now very good. She's in vet school. I'm really happy. And the other one is still a teenager, 15 years old. So that's a lot to do there, right? But I like a good book. I like, I've kind of got away from fiction now. I'm more into, I have to read a lot of leadership books, so I read those. But I also read a lot of nonfiction. Right now, I'm, all those, what do you call those? Mills and Boons, they're gone. I can't read those anymore. I'm an outdoor person. Like I said, I really wanted to work with Dr. Kotagama. But I'm an outdoor person. I like hiking and camping, that kind of stuff. But COVID actually, being home, we couldn't go to work. We were home. That actually drew me back. I used to really love gardening when I was in Sri Lanka, and I kind of gave it up. But I took up gardening again, being home and being able to do that. And then I also tried to learn how to grow orchids. So I had a whole collection of orchids in the summer, but now they have died. So I don't know what I did, but so I was, now YouTube is so great. I mean, you have all these lessons, so I was watching YouTube videos to learn how to grow orchids. And then my husband and I both love music. So believe it or not, we watch Sihine Kire, the recorded ones. And then I think there's another one, Kavi Dahayata Sindhu Dahayak or Sindhu Dahayata Kavi Dahayak, something like that. So we both like music and dancing. We like that. And we both of us also like a lot of plays. We are now in the Northeast, so close to New York. We love Broadway, the theater. So those are some of the things that I do. So,
Right.
Yeah. You have to have a balance, right?
Yes.
Of course, madam.
Yeah.
Thank you, Dr. Binodh DeSilva, for your valuable contribution for this interview. We got an idea about how to pursue a career in science and a lot more things. It was a great privilege for us to interview you and alumnus of Faculty of Science, University of Colombo. We wish you all the very best for all of your future endeavors, madam.
I wish you all the best too. both of
Thank
you.
you.
Yeah, very nice, very nice. Thank you.
Today we have interviewed Dr. Binodh DeSilva from Bristol Myers Squibb, New Jersey, USA. Stay tuned for another interview with an eminent scientist. Again, thank you so much, madam, for these valuable thoughts and ideas. Have a nice day.
Yeah, you too. Thank you.